This conference will now be recorded. This conference will now be recorded.
Yesterday we had finished telomere. We finished off DNA replication. We had said about what are the DNA damages that can take place and what are the repair mechanisms that can require to repair the damage to the DNA. And we had ended the class by saying what is a telomere and what is telomerases. What is the importance of the telomerase enzyme? So I'll just review a little bit about the telomeres. So here you can see with the replication. Please put your audios to mute, all of you. Ila mute cheyali students. It's already mute all. So with the first round of replication, what you have a leading strand and you have a lagging strand. So with the leading strand, the replication is taking place at the 5' end to the 3' end, and in both leading strand and the lagging strand. So in the leading strand, there is no problem, but what happens in the lagging strand is when the lagging strand is forming, what happens is when the Okazaki fragments are forming, there are several RNA, several primers will be there. So this is the primer over here onto which the strand is, nucleotides are being added over here. So this is occurring in the 5' to 3' direction. But when the last primer is there to which the nucleotide is adding, what will happen? These gaps with the primers are being removed, and the primers are being removed. The gaps are being filled up with the by the DNA polymerase and sealed by DNA ligase. But the problem comes in the lagging strand with the last primer. When the last primer comes and this primer is removed, this removed primer cannot be replaced by the nucleotides with DNA polymerase. So if now suppose this area is not being refilled with the nucleotides, what will happen? It will lead to shortening of the template. Now with each round of replication, if shortening of template continues to occur, then the chromosome will become shortened. So what is the solution for this in order to prevent the shortening of the chromosomes? So yesterday I told you about the telomerase enzyme. So now eukaryotes, they have tandemly repeated sequences of 6 nucleotides at the end of their chromosomes. So, and then we have the telomerase enzyme that is composed of the protein and RNA complementary to the telomere repeat. It binds to the terminal telomere repeat and catalyzes the addition of new repeats. And this helps in by lengthening of the chromosome. Any absence or mutation of the telomerase activity will result in chromosome shortening and limited cell division. So we will see here in the figure. So this is the gap that is left by the removal of the primer, and this is being helped by, or this problem is being overcome by the telomerase enzyme. So this telomerase enzyme has a RNA which provides the primer. So you can see the sequence here: T, T, G, 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 and G. So, and what is the sequence in the RNA primer in the telomerase here? It is complementary to that of the sequence here in this strand. So this is adenine A, A, C, 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 and here now there are 3 nucleotides. So for this 3 nucleotides, complementary nucleotides will be added here in the 3' end of the DNA strand. So here you can see it complementary to A, this is T, complementary to this is another T, and complementary to C is G. So the extension of the 3' end is occurring over here on the in the case where the lagging strand is there. So and then this telomerase will translocate further, and you can see when it translocate, this AAC is again, this is complementary to this repeats, and then further extension and translocation is repeated several times. So further extension of the nucleotide is taking place at the 3' end, so that the chromosome gets elongated. Then the RNA primer will be removed and this gap will be filled up by the DNA polymerase. So new DNA will be catalyzed by DNA polymerase and ligation will occur, or the gap will be sealed by DNA ligase. So this is how the shortening of DNA is prevented by the enzyme DNA polymerase, by the telomerase enzyme. Now the process of telomere shortening has been linked with cell death. Absence of the cell death in cancer cells is linked to the activation of the enzyme telomerase. So in cancer it has been found that the enzyme telomerase has become overactive. So this activity or the enzyme telomerase is more in cases of certain cancers like this keratosis congenita, due to mutation in the gene responsible for the RNA component of the telomerase. So because of this association with increased telomeric activity in the cancer, the enzyme telomerase has become an attractive target for cancer chemotherapy. So the drugs have been designed to inactivate this telomerase enzyme so they can induce the cell death in the cancer cells and prevent the proliferation of the cells in the cancer process. Now what happens is, much of the we have seen, I have told you about introns, I told you what are introns, what are exons, and what are cistrons. So much of the mammalian genome, it appears redundant and much of it is not highly transcribed. So what is a genome? Genome is completely the genetic DNA that is present, the whole DNA that is present either in an organism, or if the complete DNA that is present in an organelle. So if suppose in nucleus, the whole of the DNA that is present in the nucleus will become the genome of the nucleus, nuclear genome. Or if it's present in the mitochondria, the whole of the DNA in the mitochondria will become the mitochondrial genome. But altogether, the genome in the whole body forms the human genome. So when we say that most of the human genome is redundant, means that it is inactive and not a lot of area of that genome is transcribed. So the haploid genome of each human cell, it will consist of about 3 × 10 to the power of 9 base pairs of the DNA, which are subdivided into 23 chromosomes. So if you take this much of base pairs in the genome, the entire haploid genome, it contains sufficient DNA to code for nearly 1.5 million average size genes. But in a body, only about 1 to 2% of the human DNA contains genes. The rest are silent areas. So even though so much amount of DNA is present and it can code for so many genes, but we have only about, out of that, only 1 to 2% of the human DNA will contain the genes. So the rest of the area is silent. And about 1% of the DNA will also be present in the mitochondria. So when you take this into consideration, 1 to 2% of the human DNA containing genes, that means there are only about 25,000 to 30,000 protein coding regions in the human DNA. So here you can see this is a whole chromosome and these are the gene clusters that are present here, and this is the representation of a single gene which contains both exons as well as introns. And then from the gene you have the primary transcript and formation of the mRNA. So when we said that protein, we have the whole human genome is composed of some areas that are coding and most of the area that is non-coding areas. So the protein coding regions of the DNA, which are exons, they are usually interrupted in the eukaryotic genome by intervening sequences of non-protein coding DNA, which are introns. So in between the coding regions, you have non-protein coding regions, which are known as the introns. So similarly, when primary transcript of the DNA, that is mRNA precursors, are formed, which is known as heteronuclear mRNA. So a heteronuclear RNA, they contain non-coding intervening sequences of DNA that must be removed So from hRNA, only if the non-coding sequences of RNA are removed, then and the appropriate segments, coding segments are joined together, then only it forms the mature RNA. So mostly the introns will be much longer than the coding regions of exons. Now, what does this excess so much? We are saying that only a small portion of the DNA codes for the protein. So, what is the remaining? What does this excess DNA do? Some of the excess DNA they serve to regulate the expression of the genes during the development, during differentiation, and adaptation to the environment, either by serving as binding site for regulatory proteins or by encoding regulatory RNAs. That is miRNAs, microRNAs. Then some of the excess also make up the intervening sequence. About 24% of the total human genome is formed by the introns that split up the coding regions of the gene. Then another portion of the excess is also seen to be composed of many families of repeated sequences, the functions of which are not clearly defined. So from this excess DNA, what we have, other than the exons, we have areas that control expression of the genes. They serve as binding site for regulatory proteins, or they encode regulatory RNAs. Then you have the introns. Then you have families of repeated sequences. So all these are present in the DNA. Now, when we talk about introns, what is this function of the introns? What do they do actually? The function of this intron is not very totally clear, but they say that because they separate the functional domains, they serve to separate the functional domains, that is the exons of the coding, in a form that permits the genetic rearrangement by recombination to occur more rapidly. So because it's separating the functional domains of the exons, it allows the genetic rearrangement through recombination to occur more rapidly, which occurs more rapidly than all the coding regions were present together. Then this enhanced rate of genetic rearrangement of the functional domains might allow for more rapid evolution of the biological function. Then we'll come to what are the repeated sequences of the DNA. We said that about 90% of the DNA is made of non-coding sequences, that which implies that this information that most of the DNA is non-protein coding. So that means this information is never translated into an amino acid sequence of a protein molecule. So about 50% of the DNA is unique or non-repetitive. Then about 30% of the genome consists of repetitive sequences. So we are saying that most of the genome is redundant inactive. Part of it is a very small part of it, 1 to 2% is formed by protein coding, exons. Some of them are required for regulatory protein synthesis for regulation of gene expression, and some of them for introns, around 24% for introns. Then we are saying that 50% of the DNA is non-repetitive, and 30% of the DNA is composed of repetitive sequences. So they contain about 5 to 100 base pairs which are repeated many times. So what are these repetitive sequences? So the repetitive sequences, they are classified into two types: highly repetitive and moderately repetitive sequences. This highly repetitive sequences, usually they are around 6 to 7 base pairs in length, and the repetition is around by 50,000. So this highly repetitive sequence consists of 5 to 500 base pairs repeated many times in tandem. So the repetition, they said, it comes to around 50,000, and these sequences, they are often clustered in centromeres and telomeres of the chromosome, and some are also present in about 1 to 10 million copies per haploid genome. So most of this highly repetitive sequence, they will be clustered in the centromeres and the telomeres of the chromosome. And majority of the sequences are transcriptionally inactive. And some of the sequences, they also say to play a structural role in the chromosome. While this highly repetitive sequences, they are only 6 to 7 base pairs in length. This moderately repetitive sequences are larger. They vary around between 100 to 300 base pairs in length. And they are present in number of less than 10 to the power of 6 copies per haploid genome. They are not clustered, but are interspersed with unique sequences. So, highly repetitive sequences are clustered, while these are interspersed. And depending on their length, these moderately repetitive sequences, they are again subclassified into long interspersed repeat sequences, which are otherwise known as lines, or short interspersed repeat sequences known as signs. So this moderately repetitive sequence may be either be lines, long interspersed, or they may be short interspersed repeat sequences, signs. Now both these lines or signs, they are said to be appear as a sort of retroposons. That means they arose from movement from one location to another. That is transposition, or you can say transposon or mobile segments, what you say. So they are said to move from one location to another location in the form of mobile sequences. And this will occur through an RNA intermediate by the action of the enzyme reverse transcriptase. Yesterday we have seen this, that transcribes an RNA template into a DNA. Now these moderately repetitive sequences, they represent species-specific families of repeat elements, and one such very important family is one of the sign family is the Alu family. Alu family is present in about 500 copies per haploid genome. It is present in 500 copies per haploid genome and accounts to roughly about 5% of the human genome. Now it has been said that suppose if the component of the sign repeats, which includes members of the Alu family, they are capable of jumping into and out of the various sites within the genome. And the transposition of this mobile elements, if they are inserted into the gene, then they may have some disastrous results, or that may they result in some mutations. Such an example of mutation through insertion of the Alu sequence in the gene is a condition that is known as neurofibromatosis. Then we said 1% of the cellular DNA is also present in the mitochondria. So in the mitochondria, majority of the peptides in the mitochondria, they are encoded by the nuclear genes, and the rest of the rest are coded by genes that are found in the mitochondrial DNA. Now this human mitochondria, they contain 2 to 10 copies of a small circular double-stranded DNA molecule that makes up approximately 1% of the total cellular DNA. This mitochondrial DNA encodes for mitochondrial specific ribosomal and transfer RNAs, and for 13 proteins that play key roles in the respiratory chain. So whatever the proteins that you see in the respiratory chain, electron transfer chain, the 3 proteins, so they are being coded by the mitochondrial DNA. Now, some important features that are associated with mitochondrial DNA is that all the mitochondria is contributed by the ovum during the zygote formation. That means it is transmitted by the maternal non-Mendelian inheritance. So mitochondria is through the maternal inheritance. So in any diseases that occurs from that results from mutation of the mitochondrial DNA, so the affected mother would in theory pass the disease to all her children, but only her daughters will transmit the trait. So, but however, in some cases, deletions in mitochondrial DNA occur during oogenesis, and then they are not inherited from the mother. So, a number of diseases have been shown due to mutation in mitochondrial DNA. So, some of the diseases we are mentioning over here, like myopathies, neurological disorders, and some cases of diabetes mellitus, are seen as a result of mutation in the mitochondrial DNA. Then we have finished what are telomeres, what are the telomerases. how the shortening of the chromosome is prevented, and we have seen what are the intron sequences, what are the functions of the genome, the whole of the genome which is not transcribed or non-coding DNA, and we have seen what are repetitive sequences and 1% of the DNA that forms the mitochondrial DNA. Now we'll move on to protein synthesis. So the process by which the DNA directs the protein synthesis includes two stages. So they are transcription and translation. So if you see totally from starting from DNA replication, this replication is followed by transcription, and transcription is followed by translation. This is said to be central dogma of life. So we will start with transcription now. So the objective for the transcription will be to learn what are the mechanisms of transcription in the prokaryotes, to know what is the mechanism of transcription in eukaryotes, and what are the post-transcriptional modifications in both. So what do you mean by transcription? So transcription is the synthesis of RNA from DNA. That is, the genetic information that is stored in the DNA will be expressed through the RNA by producing the messenger RNA. So if you just see, we'll just briefly go through what is the structure of an RNA or how it differs from DNA. So in DNA replication, we had seen that deoxyribonucleotides take part over there. Here, RNA is also a nucleic acid. It's a single-stranded, but here the sugar ribose is present instead of the deoxyribose as in DNA. So important here is that the sugar ribose is present instead of the deoxyribose that you see in DNA. Then RNA has the base uracil instead of thymine that is present in DNA. Then RNA is short, only one gene long, whereas DNA will be long and contains many genes. There are several types of RNA. So you have ribosomal RNA, which is the most abundant, accounting for 80% of the total. Then you have messenger RNA, which accounts for only 2 to 5% of the total. Then transfer RNA, that approximately is 15% of the total RNAs, and the small RNAs that accounts for only less than 1% of the total. Then each of these RNA has their own functions. So the function of the messenger RNA is carrying the information, specifying the amino acid sequences of the proteins from the DNA to the ribosomes. Then what is the function of transfer RNA? This transfer RNA serves as an adapter molecule in the protein synthesis, translating the mRNA codons into the amino acids. Then the ribosomal RNA, they play catalytic roles and structural roles in the ribosomes. Then primary transcript, it is a precursor of the messenger RNA or ribosomal RNA transfer RNA before being crossed by splicing or cleavage. Then small nuclear RNA, they also play structural and catalytic roles in the spliceosomes. Then we always think that transcription is only the synthesis of the messenger RNA, but it is in transcription involves that all the three forms of messenger RNA, ribosomal RNA, and transfer RNA, all of them will participate in protein synthesis. So that means the DNA is being transcribed to messenger RNA, ribosomal RNA, transfer RNA. Then all are made by the DNA dependent RNA polymerase through the process of transcription. Now, not all the genes will encode the proteins. Some will also encode for the ribosomal RNA as well as for the transfer RNA. Now, transcription is selective. What do you mean by that? We mean that the whole of the DNA is not going to be transcribed. So this transcription is tightly regulated. Only 0.01% of the genes in a typical eukaryotic cell will undergo transcription at any given moment. So the entire molecule of the DNA is not expressed in transcription, and RNAs are synthesized only for certain selected regions of the DNA. That means transcription is taking place in only selected regions of DNA. The whole of the DNA will not undergo transcription at one particular time. Then what are the similarities in the process of DNA and RNA synthesis? So in DNA, replication of DNA, you have seen that the steps of initiation and elongation were there. But in DNA, you have initiation. RNA synthesis, you have initiation, elongation, as well as termination. But both of them have 5' to 3' polarity. Then both of them form large multi-component initiation complexes. And both of them, in both the processes, there was adherence to the Watson and Crick basing pair rules. That means adenine to thymine and guanine to cytosine. That base pairing rule will be maintained in both. But there are also several differences are also seen between the DNA and RNA synthesis. They are very important. So what are the differences is that instead of deoxyribonucleotides that are required in DNA synthesis, here in RNA synthesis, ribonucleotides will be used. Then uracil will replace the thymine as the complementary base pair for adenine in RNA. Then a primer will not be involved in RNA synthesis, as RNA polymerase, they have the ability to initiate the synthesis genome. So directly they can start the synthesis without the primer, which is not seen in case of DNA synthesis. And then only portions of the genome are vigorously transcribed or copied into RNA, whereas the entire genome must be copied once and only once during the DNA replication. And then there is no highly active, efficient proofreading function during the RNA transcription. So these are very important, 5 important points that differentiate between RNA and DNA synthesis. So they are requirement of ribonucleotide, uracil replacing the thymine, and then you have primer is not required. Then the whole portion of the DNA is not transcribed, and there is no effective, efficient proofreading function for the RNA transcription. So we will be, their transcription is seen in both prokaryotes and eukaryotes. So first we will start with the transcription of the prokaryotes. And because this has been best characterized in the prokaryotes. So there are 3 stages of transcription. There is initiation, chain elongation, and chain termination. So transcription in the prokaryotes, a transcription unit is defined as that region of the DNA that includes the signals for transcription, that is initiation, elongation, and termination. The RNA product which is synthesized in the 5' to 3' direction is the primary transcript. And the transcription rates may vary from gene to gene, but they can be very high. And the starting point of transcription corresponds to 5' nucleotide of the messenger RNA. So here, so now what do you require for transcription? We have said that we have talked about the RNA polymerase and the enzyme that is required. Then we have here, we can see the template strand. A template strand and a coding strand is there. And this is the RNA transcript that is formed from the, during the transcription process. So the DNA strand is being transcribed. So this is a coding strand and this is a template strand. Now the sequence of ribonucleotides in an RNA molecule will be complementary to the sequence of the deoxyribonucleotides in one of the strand of the double-stranded DNA molecule. So to which strand it will be complementary? It will be complementary to the template strand. So the strand that is transcribed or copied into RNA molecule is referred to as the template strand, while the other strand is frequently referred to as the coding strand of the gene. And it is called as a coding strand because with the exception of thymine for uracil, where thymine is changed for uracil, it corresponds exactly to the sequence of the RNA primary transcripts, which encodes the protein product of the gene. So this RNA transcript that is formed will be complementary to the nucleotides of the template strand, but it will be exactly the same as that of the coding strand, except the thymine being replaced by uracil. Then which is the template strand? Or we said there are two strands. How can we know from the two DNA, the double-stranded DNA, which is the coding strand or which is the template strand? Is it permanently one will remain as a coding strand or one will remain as a template strand? Is it the case? No, such is not the case, because you can see here is an example. This is a gene A, B, C, and D. So in gene A, one of the strand here is a template. In gene B also one is a template strand. But in gene C, you can see this has become the coding strand. This has become the template strand, and this is the coding strand. So it is not specific for any particular gene, which is that. In each gene it varies, which will be a template strand and which will be a coding strand. So the template strand for each gene will not necessarily be the same strand of the DNA in the double helix. A given strand of the double-stranded DNA molecule will serve as a template strand for some genes and the coding strand for some other genes. But the information in the template strand is read out in the 3' to 5' direction. But transcription will take from 5' to 3' direction. So for this transcription, the prokaryotes, a single enzyme DNA-dependent RNA polymerase will synthesize all the RNAs in the prokaryotes. So this RNA polymerase of the prokaryotes that is seen in especially of E. coli is 400 kilodalton in this one long, and it consists of 5 units. It consists of 2 alpha, 1 beta, 1 beta prime, and an omega subunit. So 5 subunits are present in this RNA polymerase. And it is this whole subunit together forms the core enzyme. So all these units together, they are called as the core enzyme of the RNA polymerase. Now this RNA polymerase, this whole core enzyme is often termed as represented by the abbreviation as E. This core enzyme, it will associate with the specific protein factor that is known as the sigma factor. So the sigma factor or protein factor along with this core enzyme, together it forms the holo enzyme, which is represented by E sigma. So this E will consist of all this 5 subunits, and the sigma factor is also present. So together they form the holo enzyme. Now each of the subunit of the RNA polymerase will have its own function. The most important is the sigma that recognizes the promoter sequence. So sigma that recognizes the promoter sequence on the DNA. Then you have the beta prime that binds the DNA. The beta that binds the magnesium ions and composes the catalytic subunit. Then the function of the alpha subunits, it appear to be essential for assembly and for activation of the enzymes by the regulatory proteins. So important for you to remember here is the sigma has to recognize the promoter sequences on the DNA, then only the transcription will begin. And once the transcription begins, here the binding the this beta the part of the DNA RNA polymerase that binds to the DNA is very much important in the transcription process to take place. So this beta prime and beta. Now this initiation, we said that transcription takes place in 3 this one phases: initiation, elongation, chain elongation, and chain termination. This initiation again we can divide into template binding, chain initiation, and promoter clearance. So remember, template binding, chain initiation, and promoter clearance. So the binding of the polymerase to the template DNA, that is the first template binding. So initiation of transcription is by what? Binding of the RNA polymerase holo enzyme and the promoter sites on the template strand. So now if you see on a whole, for transcription, what do you require? You require the DNA double-stranded helix, which consists of a coding strand and a template strand. then you require the RNA polymerase, and then you require the promoters. So the binding of the DNA polymerase holoenzyme at the promoter sites on the template strand will initiate the transcription. Now this binding of the RNA polymerase to the DNA when it binds non-specifically and with low affinity, and it will migrate looking for the promoter site. Now there are two sequences on the coding strand which the sigma factor of the RNA polymerase can recognize for initiation of transcription. So for transcription to begin, the sigma factor first has to recognize these two base sequences that are present on the coding strand. So what are these two base sequences? This sigma will recognize the promoter sequences, which are also known as the promoter sequences. They are the Tata box or Pibno box and a 35 prime 35 base pair sequence. These two are the promoter sites which the sigma subunit recognizes, and it helps the core enzyme to bind to the specific deoxynucleotide sequence of the promoter region to form the pre-initiation complex. So the sigma factor recognizes the Tata or Pibno box and the 35 base pair sequence that form the promoter sequences. And once it recognizes that, it helps the core enzyme of the DNA polymerase to bind to the specific deoxynucleotide of the promoter region. Now they together form the pre-initiation complex. So here you can see this is the coding strand, this is the template strand, and this is the Tata box that is having the sequence TAT. This is of 10 base pairs, and this is the 35 base pairs. This is a 35 base pairs upstream. This is 10 base pairs upstream of the promoter site, and this is 35 base pairs upstream of the promoters of the transcription site. These are the promoter sites to which the sigma factor will bind. So here you can see this is the sigma factor that is binding to the promoter site here, that the Tata and the 35 10 base pairs upstream, which is the Tata box, and 35 base upstream, which is the 35 sequence box. It will bind to that, and then once it recognizes this sequences, it will help this core enzyme to bind to the deoxynucleotide sequence of the promoter site. And this, once it binds, the transcription will begin. So then now the binding, once it has the DNA polymerase binds to that, what will happen? The RNA polymerase, once it binds, it undergoes a conformational change, and unwinding of the DNA around the transcription site takes place. And the first nucleotide, this first nucleotide almost always it's a purine, then associates with the nucleotide binding site on the beta subunit of the RNA polymerase enzyme. So once the RNA polymerase binds, conformational change takes place in the RNA polymerase and unwinding of the DNA occurs. Then the first nucleotide, which is almost always a purine, will be added, and this will associate with the nucleotide binding site on the beta subunit of the enzyme. So the initiation also prefers to bind ATP and GTP, and most of the RNA begins with a purine and 5' end. So initiation always requires energy, either by hydrolysis of ATP or by GTP. So here you can see this is how the unwinding of DNA duplex is taking place over here, and this is the RNA polymerase, this is the coding strand, this is the template strand. So the first nucleotide that will be added here in the RNA is at the 5' end. So starting is from this 5' end. As the nucleotides are being added, they will be being added at the 3' end. So this nucleotide, first nucleotide, we said it will be of purine. And the nucleotides that will be added, they will be complementary to the base pairs that are present in the template strand and similar to that of the coding strand. Then what will happen in the presence of the next appropriate nucleotide bound to the polymerase? The RNA polymerase catalyzes the formation of phosphodiester bond. So when the next second nucleotide is added, what will happen? There is a formation of phosphodiester bond like you have seen in RNA synthesis. And at the same time, when phosphodiester bond is formed, there will be elimination of a pyrophosphate. And the nascent chain will now be attached to the polymerization site on the beta subunit of the RNA. So at this beta subunit, the nucleotides will go on adding and the chain length will increase. So initiation of the formation of RNA molecule at its 5' end, then follows elongation of the molecule from 5' to its 3' end continues cyclically antiparallel to its template. So the enzyme polymerizes the ribonucleotides in the specific sequences dictated by the template strand and interpreted by the Watson-Crick base pairing rules. So pyrophosphate is always released when a phosphodiester bond is formed. Then once about 10 to 20 nucleotides have been polymerized, then this RNA polymerase will undergo a second conformational change leading to promoter clearance. So what do you mean by this promoter clearance? The sigma subunit will dissociate, completing the initiation process. So once 10 to 20 nucleotides are formed, the sigma subunit will dissociate and this will end the initiation. So once this transition occurs, RNA polymerase now physically moves. So from this one area with 10 to 20 nucleotides has formed, the RNA polymerase will move to the next segment of the DNA, where again that DNA will unwind. So will physically move away from the promoter, transcribing down the transcription unit, leading to the next phase of the process that is known as elongation. So here you can see in the figure, this is the RNA polymerase, this is the sigma, this is the promoter region where the sigma unit binds, and here the transcription will begin once the core enzyme binds. So this is the first purine nucleotide triphosphate that is being added, and then formation of phosphodiester bonds. So this will continue till 10 to 20 nucleotides are formed. Then this sigma factor will be released, and then the RNA polymerase will move to the further along the DNA strands, along the DNA molecule. So here we can see that as the RNA polymerase moves along the DNA, it will continue to unfist the double helix, exposing about 10 to 20 DNA bases at a time. So when it moves to the next one, what will happen? The previous DNA will rewind itself. So this will rewind itself and this part will unwind to continue the process of increasing the length of the RNA. So it will expose another 10 to 20 DNA bases at a time for pairing with the RNA nucleotide, and the RNA strand will further lengthen. So when this unwinding takes place of the DNA strands, a transcription bubble is formed, which is approximately 20 base pair area of melted DNA, and the entire complex will cover about 30 to 75 base pairs depending on the conformation of the RNA polymerase. So RNA polymerase, it synthesizes a single strand of RNA against the DNA template strand, adding nucleotides to the 3' end of the RNA chain. So you can see that this newly made RNA strand, as the RNA polymerase goes on moving to the next one. So once it completes this 10 to 20 nucleotides, again it will move on to the next part of the DNA. So this will rewind, then the next part of the DNA will unwind itself. So then topoisomerases are also present to participate in the transcription process. They both precede and follow the progressing RNA polymerase to prevent the formation of superhelical tensions that would serve to increase the energy that is required to unwind the template DNA ahead of the RNA polymerase. So these topoisomerases both before and after the RNA polymerase, they will remove the supercoils. Then how does the termination of transcription take place? There are two principal mechanisms for the termination of transcription, which is RHO dependent and an RHO independent process. So this RHO independent process is the one which is more common. So termination of the synthesis of RNA molecule in the prokaryote, it is signaled by a sequence. So when the RNA polymerase goes on synthesizing, it will encounter a sequence in the DNA strands, that is in the template strand of the DNA molecule. So this will be a signal that will be recognized by the termination protein that is known as a Rho, or it is a P factor. So it is this Rho is an ATP dependent RNA stimulated helicase that will disrupt the nascent RNA DNA complex. But in some cases, when we say that Rho, this is a P independent termination is there, in some cases bacterial RNA polymerase can directly recognize the DNA encoded termination signals without the assistance of the Rho factor. So without the Rho factor also, sometimes it can independently terminate the transcription. So here you can see this is the transcription taking place. This is the P factor, and this red here represents the termination sequence at the termination site. So there's a sequence here that indicates that the transcription has to end. So this sequence will be recognized by the P factor. So this moves along, the P factor will move along the RNA transcript, and it will halt the transcription, releasing the transcription chain. So how this halting is taking place? We are saying that it will stop the transcription chain. This is through the formation of a hairpin loop. Now, there are some inverted repeats that are present on the DNA molecule. The inverted repeats, when transcribed into RNA, they can generate into secondary structure in the RNA transcript. So that means they are forming a loop over here. So a formation of this RNA hairpin causes the RNA polymerase to pause, and subsequently the termination factor interacts with the paused polymerase and induces the chain termination. So wherever there are inverted repeats are present over here, these are the sequences that indicate that termination is supposed to occur. So at that point, it will form a hairpin loop over here, and that at that point the RNA polymerase will stop transcription. So this is here again another example of how a RNA stem or hairpin loops loop hairpins are formed. So after termination of the synthesis of the RNA, the enzyme will separate from the DNA template and probably dissociate to free core enzyme and free factor. With the assistance of another factor, the core enzyme then recognizes the promoter at which the synthesis of a new RNA molecule commences. So with the finishing of one RNA strand, the enzyme will separate. So either it may remain a free enzyme, or with the help of another sigma factor, it will recognize some other promoter site and to form a new RNA molecule. So here what we have finished the transcription of the prokaryotes, and I'll be showing you some one video for how the transcription in prokaryotes is taking place. So here this is a transcription cycling, starting with the template binding, chain initiation, then promoter clearance, chain elongation, and chain termination, followed by release of the RNA enzyme. So this RNA polymerase, again, it may participate in further RNA synthesis. So I'll be showing you a video now.
In bacterial cells, the transcription of a gene or group of adjacent genes called an operon is controlled by a set of proteins interacting with specific sequences in the DNA molecule, leading to the initiation of transcription and the termination of a transcript. Transcription is initiated just beyond the yellow promoter region and is terminated at the green terminator site. The sequence of the promoter at positions 35 and 10 nucleotides upstream from the transcription start site are critical to initiating transcription. In order to initiate transcription in bacteria, core RNA polymerase and sigma factor form a holoenzyme. Sigma factor recognizes the sequence of DNA at the promoter region. This holoenzyme then binds to the promoter region of the DNA molecule, forming the closed complex between RNA polymerase and the DNA molecule. By breaking hydrogen bonds between bases in the double helix, the closed complex is converted into the open complex. The formation of the RNA transcript from nucleotide triphosphates then takes place as RNA polymerase is forming the new transcript, starting at the 5' end of the transcript. New nucleotides are added onto the free 3' end. The initiation of transcription is completed when sigma factor is released. As elongation of the transcript then continues, the RNA polymerase moves down the DNA molecule and the next sequence in the chain is opened up. As RNA polymerase moves down the DNA strand, the DNA molecule entering RNA polymerase unwinds, forming the open complex, while the DNA exiting RNA polymerase rewinds to form double helix. Ribonucleotide triphosphates are added to the chain, and the growing RNA transcript continues to elongate as new DNA sequence enters the open complex region. The new RNA transcript initially forms an RNA-DNA hybrid, complementary to the template strand, corresponding to the sequence of the coding DNA strand. In this way, the DNA double helix is opened, transcribed, and reclosed with minimal stress on the DNA molecule. One mechanism of bacterial transcriptional termination involves the binding of the Rho protein to the RNA. It is a helicase that moves in the 5' to 3' direction toward the RNA polymerase. Between the Rho recognition site and the terminator site, a stem loop structure is formed by the sequence in this region. This stem loop structure halts the movement of RNA polymerase, and the Rho protein then moves to the RNA polymerase complex, causing the complex to dissociate and terminating transcription, resulting in the full-length mRNA transcribed from the gene.
So were you able to understand the whole process? It's a very beautiful depiction of in the video. So any doubts you have?
No, ma'am.